Today I'm going to talk about one aspect of my research. I have a kind of a just as amphibians are biphasic. I have kind of a biphasic life in my research. I spend part of my time working on issues that, for lack of a better term, have to do with rapid evolution, and I'm going to talk about two specific projects today. One from a few years ago, beginning a few years ago, and one more recent. Another aspect of my work has to do with disease ecology, and I've met folks interested in both sides of that today. It's been a lot of fun to talk to people. So what I'm going to do is run through first a project that I started several years ago, trying to understand whether ecologists need to worry about evolutionary dynamics. And then I'm going to talk about a project more recently that we've got into that builds on some of these themes. Anytime a card-carrying ecologist comes into your department and starts telling you that they're going to tell you something about evolution, you should kind of look for your wallet and make sure that I'm not trying to do something bad here. I'm not going to claim to you that I'm an evolutionary biologist by any stretch of the imagination. The way I got into these topics is because I was kind of confronted with situations, I believe, where I couldn't ignore evolutionary dynamics and understand the ecological aspects of the system that I work in. The mindset that I kind of came up through the ranks with is the paradigm that many of us in ecology have held for a long time. And this concept has to do with notions of ecological time versus evolutionary time. And I'm not trying to pick on Larry Slobodkin here, but he phrased it most clearly. And so he gets quoted quite a bit about this, but I think he was reflecting prevailing attitudes when he said that ecologists don't need to worry about evolutionary dynamics when they're studying the phenomena that they're interested in, because more or less over scales of, say, 10 generations or so, we can expect steady-state dynamics in terms of, say, the effects of whatever genetic changes are going on the phenotypes that may influence ecological patterns. Whereas evolutionary time takes place over scales of hundreds of thousands of generations. More recently, this idea has really been taken to task as missing a whole bunch of information, some of which was available at the time Slobodkin was writing, but by and large came around as a product of the explosion of work in both ecology and evolution that really began in the 1960s. And Nelson Hairston Jr. and colleagues did a really nice job of summarizing this viewpoint in a paper from a couple of years ago called Rapid Evolution and the Convergence of Ecological and Evolutionary Time, picking directly up on Slobodkin's idea and showing, in essence, that there have been a number of studies that have documented rates of evolution of ecologically important phenotypes sufficiently fast that they have the potential to impact the outcome of ecological interactions while they're underway. And that they used that as a definition for what they thought we should say rapid evolution is. There's a lot of, as with a lot of terminology in our disciplines, there's a contention about whether we should say evolution is rapid or not, ever or when or whatever. I'm going to sidestep that a little bit today. I'm going to tell you that the kinds of evolution that I'm seeing are ones that I wasn't aware of until I started looking for them, and now that I see them, I realize that they're probably fairly important for the things that I study. If that's rapid evolution, great. So I'm going to start talking about a pattern that I've been interested in since I was a graduate student, and that is the influence of the vegetation surrounding wetlands on what goes on inside of them. Very briefly, the world can look like this. This is actually a beaver wetland in Connecticut where I do much of my work. This used to be forest. Beaver dammed it somewhere over here. All the trees were killed, so there's no overstory over this body of water, and there's lots of sunlight flooding in there, versus places that look like this, where, for whatever set of reasons, the forest has grown up around there, and there's just little patches of sunlight that are reaching the surface, either as flecking in here, or you get little concentrations of light. We've attacked this by doing work to estimate what the light environment is like in these places, and what the consequences of those patterns might be. So where we're coming from is the knowledge that most amphibians use very small wetlands, and these are easily overtopped by overstory trees. So your average hardwood tree has a canopy diameter that can easily span half of a basin of the places where most frogs live. Wetland canopies exist in a variety of successional states, and they can change really pretty rapidly. Certainly, I'll show you some agents of change that can remove canopy, but even when we're talking about the growth of canopy, 20 years is enough time for this to change. Some of the work that got me interested in fact, the work that got me interested in was going out and resurveying the work of a person that many of you know well, and that's Henry Wilbur. So Henry is a graduate student at the University of Michigan in the 1960s, started surveying ponds that we started surveying about 20 years later, and the canopy conditions had changed dramatically to the extent that Henry, walking into these places, sometimes couldn't recognize them as the same places, and that's the excuse he used, is that they had changed a lot. Okay, and finally, wetland canopies strongly affect water temperature, and so we've been using this as a system for studying response to thermal change. So what changes wetland canopy? There are a couple of things that have been around for a long time. Beaver were once very common in most of eastern North America. They were wiped out in southern New England by about 1800. But since about the 1950s, 60s have come back and are now very common in parts of Connecticut for sure. We also have windstorms. People in this part of the world don't need to be told about what windstorms can do to forest areas. In particular, in our part of the world, they hit less frequently but still have devastating effects. So this is one of the small towns near the Yale Forest where I do my work, and that's the aftermath of a hurricane in 1938. That we can still see the signal of in the vegetation today. At the Yale Forest alone, a 7,500-acre area, about 10 million board feet of merchantable timber was knocked over by this storm. Okay, canopy temperature. When I talk to plant ecologists, forest ecologists, this is the duh slide. And they can't believe that it took wetland amphibian people about 100 years after the forest people to figure out that something like this may be going on. Nevertheless, I ask you to bear with me for a moment. On the x-axis here, we borrowed some forest ecology technology by taking these hemispherical photos across a 5-meter grid inside the wetland basin. If you're in a totally black box and not one photon can get in there, your global site factor, or GSF, is 0. If you're out in the middle of a sunny plain with nothing blocking you from the sun, it would be 1.0. And across a set of wetlands, you can see very strong relationships between global site factor and temperature. One thing that maybe people can't appreciate when they look at this temperature, particularly when I'm down in nice sunny warm places like this, they look at 13 or 16 and they go, that's all cold. But if you're a little wood frog tadpole, this could constitute a doubling in growth and development rates across that gradient. Okay, so here's the line of thinking that I'm going to use in evaluating what this means for amphibians and whether we should see evolutionary response to it. Canopy cover, as it increases, should lead to decreased development rate. As temperature goes down, development rate of these ectotherms is going to go down. Development rate, for reasons I'll elaborate on, is likely to be under strong selection. And I'm going to predict that there's going to be counter-gradient variation in development rate. And some of you will know what that is. For those of you who don't, I'll do a couple slides on this. Counter-gradient variation is geographic variation in genotypes that counteracts an environmental influence across a gradient in a manner that diminishes phenotypic variation. Got that? So here's a gradient, and here's a phenotype, and you can see that it varies across this phenotype. What counter-gradient variation does is tend to put pressure that flattens this out. So in the context that I'm going to be thinking about this, as temperature goes down, development rate goes down. But the development rate going down is a bad thing if you're a little tadpole living in a pond that's going to dry up. And so there's going to be selection, we're positing, that's going to tend to try to flatten that out. Okay? It would be better to develop faster in a cold environment because all else equal, those ponds are going to dry up about the same time as the ones that are in the sun, and you're going to be developing a lot more slowly, and so you could have mortality as a certain endpoint. And we've got some evidence that this does go on in amphibians. This is one of the most famous frog papers of the 1970s. And when I say that to my students, they laugh because they think the idea of reading anything written in the 1970s is somehow some esoteric, effete exercise. But this is a very important paper done by Keith Bervin, Doug Gill, and Sandra J. Smith-Gill, where they did some very nice work in showing what at the time was called countergradient selection, what we more conservatively call now countergradient variation, existed in green frogs that lived across an elevation gradient in the Blue Ridge Mountains, going from montane areas up above 850 meters compared with lowlands. And essentially what they saw is out in the field, green frogs in lowland environments can get out in as little as 3 months. But when you go up onto the ridge tops, it takes at least a year. Okay? Now when they take embryos out of these populations, bring them into the lab and stick them in an incubator, the larvae from the montane populations actually develop 26% faster. Okay, so even though the field pattern runs in the opposite direction, the idea is they didn't do this, but if they had done transplant experiments, what they would have expected to see is the animals from the montane populations in the montane environment develop faster than the ones from the lowland would have. And this they took as evidence that these populations had evolved more rapid development rates to cope with this. Okay. So I asked a related question, and that is, does countergradient variation occur over small scales of space and time? So when this pattern came out, people thought that maybe this is a very old thing, that it took many, many hundreds, if not thousands of years for that kind of a gradient to set up. But what we were seeing with these canopies in one pond versus another is we were getting the same degree of temperature difference in two ponds that might be from here to the back of the room apart, because one's got sun in it and the other is totally shaded, as what they were seeing from the ridge tops down into the valleys. And so I asked the question, essentially, can we expect frogs to evolve rapidly in response to altered thermal environments over these very small spatial scales? And the temporal scales may be thought of as shorter as well, because we know from aerial photographs and from the timing of the introduction of beaver that the canopies can change over much shorter time scales as well. So the result was this paper that I'll tell you about right now. So this involved first going out into these ponds, and here's one of them, and estimating global site factor across the pond. So here's a smooth surface of this pond, and you can see here's this big light spot in the back. That's right here. And frogs are not bright. I was touring around with Anne yesterday looking at the lemurs, and they're certainly not as bright as these video playing lemurs, but they're not that dumb. And if you go out into these ponds, this is where wood frogs lay their eggs. So they can find these sunny spots. Nevertheless, the water temperature in this heavily shaded pond overall is pretty low. So we do this for a set of a dozen ponds or so. Now I want to set up just a little bit about the biology of these things. So the reason that we're positing that embryos and larvae of wood frogs are going to be under strong selection is because in a year of typical rainfall, this is the scene in about a third of the ponds. So the ponds dried up before anybody's gotten out, and you get this mass of wriggling tadpoles that feed the snakes and the ants and stuff, but don't end up ever getting a chance to turn into frogs. It's a very sad scene. So the study approach I'm going to take here is two-part. First, in situ observations of embryonic development, just how fast do embryos develop in the field. We're going to do that in 6 natural wetlands that vary across this canopy gradient. And the way we're going to estimate this embryonic development time is the time between the first day that we see eggs and the first day that we see hatchlings in the water. Common garden experiment will follow on from that. We're going to use 12 wetlands that vary in canopy. We're going to estimate embryonic period. I'll have more to say about that. In temperatures, in incubators that are set to temperatures that kind of span the range from the warm ponds to the cooler ponds. So for this experiment, we're going to collect recently laid eggs. So basically, we had to camp out in these ponds, and as the eggs came out of the frogs, we were gathering them up and bringing them immediately back down to New Haven to get them into incubators. So we were collecting 12 eggs per mass, and a total of 6 masses from each of these wetlands. We used tissue culture plates that have 17 mil wells and put 1 egg into each one of them. assigned the plates to high or low temperature incubators, then monitored eggs until hatching. So what we end up with is a design where we've got 12 different wetlands, 2 different temperatures in the incubators, and we're asking, does embryonic period increase with global site factor? If it does, then we have evidence of this countergradient variation. So these are aerial photographs that have been stitched together that cover the Yale forest in northeastern Connecticut. This is an area that the school has owned since the 1930s. It was largely agriculture. You can kind of see the remnants of that. There's not much of that left these days. It's mostly forest cover, and it's about 7,500 acres. And so the wetlands here are spread across here. They have in common that they're all annual temporary wetlands. They all have wood frogs breeding in them, and we, even before we started this, we had several years of information on these places, so we knew that they reliably had wood frogs. Okay, so what we're doing then is building off this idea that there's a gradient of wetland temperatures that relate to global site factor. We actually estimated the thermal environment inside of these egg masses. So this is what wood frogs do. They're very friendly animals, and when they're going to breed, they all get together at one time in the year. I know when this is because my neighbors tell me they hear the ducks are quacking in their backyard, but it's actually wood frogs. And so you can find hundreds or even thousands of eggs that are all in one spot. This is amazingly convenient if you're doing any kind of population estimation, as we also do, because you can get an estimate of female input into the population because each female lays no more than 1 egg mass per year. So this gives you an idea of how many females bred in a pond during the year. Okay, so what we're doing is collecting these things at as early a stage as possible, getting them into the incubator as soon as possible, monitoring development, and trying to capture the moment when they actually hatch out of the egg. So all these photographs are actually from well plates documenting these different stages. So we would pull these things out every couple of days, stage a plate very quickly, and get it back into the incubator. So when we went out into the field, we saw this. Again, this is one of those things. It's not very surprising. But as we go from heavily shaded places to open canopy places, we get a decrease in embryonic period. That is, the sunnier it is, the faster these things get out. So we've actually got a range from 18 days down to 9 days. So that's a big deal. This is the difference in many cases between happy little metamorphs hopping out of the pond and the sad piles of dying metamorphs. It's often that close. If the pond, if it rained and it got a week more, they would get out. And then this is what we found in the incubators. So just without even getting too sophisticated about this, we have a negative slope, and then with the same axes, we have positive slopes for low and high temperature. Okay? So the idea here is that as we go from low light environments to high light environments, we're actually seeing an increase in embryonic period. So this is the same, qualitatively, this is the same thing that Vervin and colleagues found over big elevational gradients in the Blue Ridge Mountains in the 1970s. We're seeing in congener in spatial scales that go on just kind of meter scale. So what we took away from this, well, the results are compared with open canopy tadpoles, closed canopy tadpoles develop more rapidly in a common garden. And we're saying that this is consistent with rapid localized adaptation, a countergradient variation pattern. I'm well aware from my evolutionary biologist colleagues that this doesn't reach the standards for demonstration of these sorts of things for fruit fly type folks. But I'm not a fruit fly type person, and I'm an ecologist, and I think I should get some credit for even going this far with it. But what this means to me is that when you see a phenotype in the field, the way that I thought about my system from the time I began graduate school until I did this work was that, at this kind of spatial scale, a wood frog is a wood frog is a wood frog. And the way I actually would stock enclosure experiments is going and getting a wood frog and a wood frog and a wood frog from all these ponds and mixing these things together, and then looking at responses where I've sampled across genetic variation in a way that I thought was responsible ecology. And now what I think I was doing was creating these horrible chimeric assemblages of things that were behaving perhaps significantly differently to the environment, from each other in ways that reflected their geographical heritage, where they came from. And so it certainly changed the way that I do my work and the way that I interpret things in thinking about ecological patterns. One of the follow-on projects I did in collaboration with my wife, Kayla Friedenberg, who is a research scientist at the Forestry and Environmental Studies School, was a chance to do spousal science, and we came through this with flying colors. We still talk to each other and everything. We did this project together looking at thermal preference behavior in the same species across some of the same ponds that I've just described to you. So this project was based entirely on natural history observations. How much do ecologists get to say that anymore? Where my wife was working in these ponds, and she noticed in ponds that looked like this that wood frog tadpoles were aggregating in sunflecks, to the extent that as the sunfleck tracked across the pond during the day, the aggregations would follow the sunfleck. And she got a thermistor and measured the water temperature, and it was just the water temperature, not the temperature of a body sitting there in that water. A couple centimeters below the surface could be 2 or 3 degrees C higher, which again is a big deal for an ectotherm, a little tadpole in these conditions. And so we saw that behavior in these ponds. We typically did not see aggregations in the open canopy ponds. No one ever had seen them. So we decided to see whether the thermal preference trials could give us some insight into whether that pattern had a basis in the kind of behavior we could measure under controlled conditions. So we picked out 3 open canopy ponds, 3 closed canopy ponds. These are 6 of the 12 ponds that I talked about in the last experiment. Collected eggs, again, right out of the animals. Those of you who know amphibian biology, when you see eggs like this is minutes away from when they've been laid. Put them into an array of kiddie pools so that they all had a common experience before we put them into thermal preference trials, and then take 10 of them, batches of 10 of them from individual populations, and put them in thermal preference arenas. And I was too embarrassed to show you a picture of the actual setup because it's just plastic gutter from Home Depot, so this looks much more official. But these are about 75 centimeter long pieces of plastic gutter. One end is on top of an ice bath. The other end is on top of a hot plate, and we're getting gradients of about 10 or 11 degrees C down here up to about 30 degrees C here, and very nice roughly linear change in temperature in between these. And so we would pair up populations and put the animals in there, let them acclimate for about a half an hour, and then film them for an hour when nobody was in the room with a camera overhead. And then poor Kayla had to look at all of that film. And so what we found by binning the temperatures into these aggregations that allowed us roughly equal numbers of observations in each bin is that with the open canopy tadpoles, there was no tendency for them to aggregate in one end or the other. So this is not different from what you'd expect if they were choosing their location at random. Okay, we couldn't distinguish this from just tadpoles cruising around doing whatever they wanted, irrespective of temperature. The closed canopy ponds, remember there's 3 of each type, so that's where the error bars are coming from. They showed a very positive signal relative to temperature. So they were aggregating at the warmer end of these thermal gradients, and that was consistent with the idea that perhaps the patterns that we were seeing in the field were the result of differences in thermal preference behavior. And we were very anxious after we saw these embryonic development results I talked about in the last experiment to look at a totally independent trait to see whether we could see some signal of the same sort of thing. We've also done work now on critical thermal maximum. That involves cooking a tadpole till it's not going to live anymore, so I'm not going to tell you much about that, except to say that critical thermal maximum also shows a pattern that differentiates between what we've defined as closed and open canopy ponds. So now we've looked at 3 different traits, and we're seeing evidence of the same thing, and that is that there's been differentiation that's related to canopy in these places. So, summarizing these results, compared with open canopy tadpoles, closed canopy tadpoles were predicted to be more temperature selective and to prefer warmer temperatures. We were able to find evidence in support of both of those. And again, we're interpreting this as being consistent with rapid localized adaptation. And this is, again, a counter-gradient pattern. because what these things are doing is tending to minimize the difference in the temperature that they experience relative to the gradient that's available if they weren't paying attention to temperature. All right, now for part 2. This is a more recent project that was my personal experience that has convinced me that it's worthwhile to go to meetings. So I had the opportunity to go and give a lecture at the Australian Society of Herpetology meetings a few years ago, and one of the talks that was given was by Ben Phillips, the first author on this paper. And this wasn't yet published at the time, but they were finding that there was evidence that cane toads in Australia, where they had been introduced in 1935, were evolving longer relative leg length and moving more quickly. And so I'm going to step through this was just one of those one-page Brevia papers in Nature. So I'm going to step through the 4 panels of this figure and then tell you about what we did as a follow-on to that. So what they found is that when they radio tracked toads, the ones with the longer relative leg length actually moved farther. Okay, so being a relatively long-legged toad seemed to be related to mobility, and that longer-legged toads were, this is the order of arrival from first to last, and this is relative leg length. So the ones that get there first at the invasion front have longer relative leg length. One of the interesting things they also found is that relative leg length declines with time since colonization once the population was established. And you can see there's lots of points here. These are many, many different sites that they've sampled. And comparing time windows from 1945 up through 2005, it looked like the invasion of toads across the Australian outback was moving faster and faster over time. This last observation, this was all really cool, but this really caught my attention because I had postdoced in Australia, and I remember in the early 90s when I was there that people were saying that the cane toad had just about reached the limit of their bioclimatic envelope and they were going to stop moving. And here was evidence that, in the time since I had left there, they had started moving faster and faster. And of course, the case that these folks are making is that this was perhaps somehow related to some of the trait changes that they were seeing. So in collaboration with Ben Phillips and Rick Shine at the University of Sydney, Mark Urban, a former student who's now a postdoc at NCEAS, and I, we were the modeling geek squad in North America, and I don't need to tell you what kind of folks these are. These are hardcore herpetologists. This is a brown snake, which is one of the most dangerous snakes in the world, and this is what Ben Phillips does on his free time. So what we wanted to do was revisit this idea of where cane toads were going to end up and see whether there was evidence that this rapid evolution that these folks had documented was going to manifest itself in either these traits or other traits that might have changed in where these things were going to end up. Most of you will have heard of cane toads. Certainly the Australians consider them one of the most notorious invaders in the world. Probably the biggest issues that arise from cane toads is that they're toxic to predators. They're toxic to the native predators like those snakes. The first work that Shine and company did was to look at effects of cane toads on their beloved snakes, because they're a snake lab, that's what they study. And one of the most amazing things that they found was that snakes were evolving smaller relative head size after the toads came through. And the dose of the toxin that they get is correlated with the size of the toad they can eat. The size of the toad they can eat is correlated with their head size. So we're going to end up with microcephalic giant snakes as a result of cane toads coming through. They are themselves voracious predators. They have a very high reproductive rate, and they're good dispersers. This is Toadzilla. He was discovered right around the time Hogzilla was discovered, so they called him Toadzilla. So that's the largest cane toad that's been found in Australia. And this is a very famous video. Hands up if anybody's seen cane toads. So I don't need to say too much, except that's a pretty funny movie. A friend of mine knows this woman. She's now all grown up and is still known as the cane toad girl. Okay. Cane toad invasion dynamics. 2 questions that we want to ask in this project. First, has the cane toad's bioclimatic niche changed? Okay, I'll have more to say about that. But we're going to be doing something different from what the prior folks have done. The standard in this arena is to base a prediction of where a species is going to end up on the native range. That makes a lot of sense because when a species is first invading, that's really the primary data you have. And it also is based on this idea of niche conservatism, which is the idea that when you move a species around to a new place on the Earth's surface, its bioclimatic envelope that determines the boundary of its range in its native area is going to be the outside boundary of where you're going to find it in the new place. Now, that's not just an academic notion. That is the notion that's used by just about every agency in the world that's mandated to decide for intentional introductions whether this one goes in or you say no. And this was an intentional introduction. And so we're going to ask, has it changed? And we're also going to go back to this dispersal rate question for a moment. Some of you will know the story of cane toads in Australia. They're native to northern South America and a big chunk of Central America. The blue here is their native range. They've been introduced across the Caribbean and in South Florida. And they've been introduced in other parts of the world as well. Notably, for our purposes, Hawaii, where there was a big cane industry. They were introduced to control the grayback cane beetle. And the Hawaiians convinced the Australians that they were a raging success, and we can send you for a very modest fortune some of these cane toads to solve your problems. And in fact, the cane toad didn't really do anything to control cane beetles in Hawaii, and it did nothing to control them in Australia. They were introduced to Gordon Vale, Australia, here on the Queensland coast, and they did very well, but they had no biological control effect at all. The toads are nocturnal, the beetle is diurnal, the toads are on the ground, the beetle's up off the ground. I mean, it was just really not, it wasn't going to work. My wife's part Hawaiian, and I've said cast some aspersions on Hawaiians that she doesn't like to hear because of this. All right, so the model that we're going to use is a logistic regression model. What we're going to be doing is using the kind of predictors that people have thought about in the past. That is climate-related predictors like temperature, so that's maximum, minimum temperature, annual precipitation, a moisture index, and evaporation index for each minute-by-minute grid cell across the Australian continent. Topography, so what is the average elevation within a minute-by-minute cell? What is the heterogeneity, so the difference between the maximum and minimum elevation? And then what we added that people hadn't messed around with much before are human-related variables such as the road density and the degree of development, which in the Australian Geographic Information System databases that we could get was an urban cover index. We used Australian data because by 2005, when we started messing around with this, there was plenty of Australian data going way back, almost from the very beginning when they were introduced. People kept track of them, first because they were excited to see them come, because they thought they were going to make everything better, and then later because they were really angry that they showed up because they were going to make everything worse. And so we have presence data, and we even have a decent amount of absence data, because once government ministries started getting into the picture, they started surveying point localities and making determinations about whether the toads were present or absent. So we actually have absence data too, which helps tremendously in these sort of logistic regression approaches. And so what we did was use these predictors to predict these patterns and use different combinations of these variables and took the top 20 models using Akiyiki information criteria and developed average regression coefficients out of those to develop an overall kind of best model that we could come up with. And then plotted presences wherever we exceeded a 50% likelihood threshold that the species was going to be present. These are more or less well-accepted techniques for using logistic regression to do bioclimatic modeling. The difference for us is that we had a lot of data, and we were able to resolve these predictors on a much finer scale than had been done before in Australia. So here's a picture, kind of a color map of the invasion of Australia by cane toads. And what you can see going from blue through the reds is how recently they arrived. So you can see for a long time, they were introduced in 1935, so that's the darkest blue. But it stays blue quite a ways out here. So this gave people the impression that these things weren't necessarily going to get very far, because even by 1960, they were fairly well contained. And then starting in the 1970s and accelerating through the 1980s and past the millennium, they started moving faster and faster. And I'll show you another map of that later. So some of you have been to Australia, some of you haven't. To give you an idea, this is a map of average temperature across the continent. I want to just give you some pictures of where these things started and where they're going. So in 1935, they were introduced to coastal Queensland. This is a very wet environment. So the entire Queensland coast has very lush forests. It peters out pretty quickly away from the edge, but this is where the sugarcane agriculture is in these very, very wet areas. As you head south, it dries out a little bit down here, and so they reached Port Macquarie down here on the New South Wales coast just a couple of years ago. They reached the Gulf of Carpentaria up here in the 1980s. And this is a picture of Ben Phillips in that part of the outback, and it's very dry up there. And they got to Timber Creek, right near the Western Australian border, in 2006. And again, this is extremely dry environments. These are places where people, by and large, didn't think the toads would get. So one of the things that we began with as a starting point is that people had tried to do this exercise, this exact same exercise, using the traditional approach of basing the bioclimatic envelope on the native range. And this is the map that they came up with. So the contiguous range was believed to be this area from just south of the New South Wales border here, all the way up to the tip of the Cape York Peninsula, more or less where it's wet. And then it was believed that they would have to be transported around the Gulf of Carpentaria. And then the top end here is also very wet. This is where Kakadu is, and there's rainforest up here. It was believed they would have to be transported up there, and if they got in there, then Darwin could have cane toads. And in fact, Mike Tyler, a very well-known Australian frog biologist, wrote a book in the 1980s where they talked about how, unbelievably, some school teacher up here in Darwin ordered some cane toads because the lab frog in Australia is cane toads. So when high school kids dissect frogs, cane toads are disposable. Families make, Saturday night in Queensland is going out in your yard, filling up a pillowcase with cane toads and putting them in the freezer. You get to drink beer while you're doing that. That's something that families do, and that's an environmentally responsible thing. That's like shopping at Whole Foods for us. Somebody shipped some up there. This guy left them in an open cardboard box in his garage, and the box tipped over, and 20 cane toads got out. And so Mike Tyler's down here in Adelaide, and he gets the toad hotline call that they've got to go race up there and catch all these cane toads. And so they did. And they got, I can't remember, but it was in the teens. They got most of them. And they didn't know how many males, how many females. Everybody was freaking out. But they didn't establish them. And this was the late 70s, early 80s. And they thought they dodged the bullet because this is the map they were thinking about. Okay. So this is the same approach that we used. We have more finely resolved data. We have better GIS than they would have had then. But this is the map we came up with. They use Central America, South American data to say this is where they're going to go. Based on data up through 2005, this is where cane toads are predicted to go. So this is an area roughly 3 times the size of what this earlier prediction said. It involves 76% of the coastline and virtually all the major cities in mainland Australia. There are some places that offer potential blocks based on this model, but we're imagining that this map can change in the future too. And we don't think the bioclimatic envelope is going to get smaller. So we're already at a point where Melbourne, Adelaide, Perth are all expected to be hospitable places, and also the surrounding areas as well. Now, for a minute, I wanted to talk about the dispersal rate. So instead of plotting when they got here, this blue to red map is now showing the rate of radial expansion of the range. And what you can see is for a long time, the rates were, this is kilometers per year, they were 25 or 30 kilometers per year or less, and it was much less in the beginning. And this is this sprinting that Ben Phillips et al. Documented in that Nature paper. And so we went back at this and developed some models that I don't have time to go into. But what this suggests is that this acceleration is real. So the conclusion from this part of our work is that the niche of the cane toad is not conserved. There's no evidence of this niche conservatism that's the basis of intentional introduction management. We also find evidence for region-specific dynamics. And by that I mean that this is an invasion front too. So they're moving this way. They're also moving this way. In those areas, the invasion front is moving very, very slowly. But there's a population up here, and this is the region where the longer leg length and so on was picked up, where these things are accelerating. So it looks like we have a case where we may have rapid evolution going on. Now I want to take just a minute or two. I know I want to finish up soon, so there's some time for questions. But in addition to these region-specific invasion dynamics, I want to touch on a larger issue that I think this is relevant for. And just to note, not to make these things too unappealing, but one of the things that cane toads in Australia love to do is sit on fermenting dung piles. And so the best place to find groups of cane toads is sitting on a cow pie. I think the work that I've talked about, both the first and second projects that I've outlined here, have implications for something that's on a lot of people's mind, and that is climate change and what we should be expecting. So a bunch of colleagues, including some Australian folks and some people who are card-carrying real evolutionary biologists, got together and just to make the point that everything we understand about how organisms respond to large changes in their environment suggests that while you don't want to get in the business of guaranteeing that any given species is going to evolve, neither should we be expecting that they're not going to evolve. And what prompted me, I'm just going to skip over the acknowledgement slide for a second. Oh, come on. What prompted me to get into this is I was a postdoc in Peter Creeve's lab around the time that this book came out. And one of the chapters in there was Evolutionary Genetics and Climate Change. Will animals adapt to global warming? In 1993, the answer to this question was maybe. A few years ago, C.D. Thomas et al. Came out with a paper that got a huge amount of play about about climate change being a risk to millions of species. And the answer to the question, is evolution going to play a role here, went the other way, even though the evidence that species can evolve rapidly had gone in the exact opposite direction. And this is particularly ironic to me because one of the 4 parts of an IPCC report is adaptation. And if you go into the social science and policy world, there are whole rafts of social scientists who are thinking about adaptation. What they mean by adaptation and what biologists mean are certainly different. But it's a subject that they think and talk about a lot. Whereas those of us in the non-human biology world, this is kind of a taboo thing that hasn't really been talked about. But it is going to be a big part of the response, I think. If you hit a population large enough to drive it to extinct or make it nearly extinct, we should also expect that there are going to be evolutionary implications if the species is able to survive that. And that's still a big question. And now I will backtrack, sorry about this, to my acknowledgement slides. That would be bad if I didn't show you my acknowledgments. So this work was based on the work of lots of folks and some very generous donors and funders. So with that, I will finish up, and I'd be glad to take your questions.